ever revision stream. It's kind of mad that I can finally say that now. So for those unaware, I started doing these revision streams all the way back in year 11 for GCSEs. And yeah, now we're here after 3 years of revision streams. We are finally at the end. Tomorrow is my last exam, and then that's me done. Which is, again, it's mad to think. And what I'm hoping to do in this stream is just spend maybe an hour going over some paper free stuff. I don't know if there's much I really want to revise, more so just little bits that I want to kind of keep in my head. So that is the plan for today's stream. And yeah, I'm kind of looking forward to just being able to, put this all beyond me now. All of the revision streams. I mean, yeah, I'm doing university and it's probable that I'll have to be doing revision for that. I mean, yeah, but I don't know whether I'll be doing university revision streams. That is not something I've considered yet. Anyways, let's get on with this. So, what we were doing earlier today is I looked through every one of the past papers, and I was looking at the essay questions that they could ask, and we just kind of did generic plans for them. And I think that's probably what I'm going to continue doing as well. Let's take a look. So, obviously the whole thing with paper 3 is its accumulation of every paper, content-wise. I want to look at the required practical stuff in particular, because we've only been asked one practical so far between papers 1 and 2. So it's very likely we're going to get asked a practical-based question today, tomorrow. So I'll go to the 2022 paper. And we'll literally just go look for any practical content. Okay. So, I am seeing a question on... Oh, okay. Interesting. This is the mitosis practical. This is what I'm seeing here. The practical where you have to investigate the rate of mitosis. That's an interesting one, because I don't recall what we got in terms of mitosis or cell replication questions in paper 1 and 2. And it gives us the method here. It gives us a method. Cut the ends off, place in hydrochloric acid, rinse them, put them in a microscope, add your stain. Okay. Suggest why the student soaked the root tips in hydrochloric acid. Okay. So obviously in this practical, you are using the tips of the roots, because those are the meristem regions, the regions where the plant can actually undergo mitosis. That's why you would use the very tips. You would soak it in hydrochloric acid because that's going to break open the cell walls. It breaks open the cell walls, allowing the stain to actually enter. So allowing cell. I don't know whether cell would be a bit of a vague term, but allowing cell to be stained. Also, you would add hydrochloric acid because that would stop mitosis. So, so you're actually able to observe it. So that is why. If I can find the mark scheme. Yeah, So to break down the cell wall or hydrolyze the cell wall, allowing the stain to pass or diffuse into the cell, or to stop mitosis. Okay, so could have elaborated there. Allowing the stain to diffuse into the cell. Okay. Fairly obvious, but that makes sense. Okay. Next question. Pressing the cover slip downward enabled the student to observe the stages of mitosis clearly. Explain why. So this is where you press the cover slip down and it basically like flattens out the cells. I'm assuming what they want you to talk about here is the fact that it's creating a thin layer of cells. So it prevents overlaps. So you can observe the cells. I'm assuming that's what they want you to say. Yeah, to create a single layer of cells. Okay, second mark point so that light can pass through. That's a mark point I always miss. But yes, so that light can pass through. Okay. Then it's asking you to describe stages of mitosis. This is something I might fully go over quickly. Mitosis is kind of one of those ones where there's a lot of minor details that you can forget. So basically, you have interphase, which is, not actually part of mitosis. The growth stage where DNA replication, organelle replication happens there. Prophase, the thing is, I think you can tend to overlap the stages ever so slightly. Prophase is where the nuclear envelope breaks down. You can tell when a cell is in prophase because the chromosomes are condensed and they become visible. This is also where you have your centrioles move to opposite poles. Or the spindle move to opposite poles. Then metaphase is where the chromosomes line up at the middle of a cell. Or the equator, sorry. This is where the oh my god, so many words. The spindle fibers are released from the centrioles, and the spindle will attach to the centrioles on the chromosomes. No. Oh my god, I'm getting the words mixed up. The centromeres, I believe. The centrioles release the spindles. The centromeres attach the chromosomes together. What else happens in metaphase? I'm assuming I've said everything. Then in anaphase, that is where the chromosomes are pulled apart to opposite parts of the cell, opposite sides, I'm assuming that you're allowed to say. Anaphase is where the centromere divides. And this is where you get your V-shaped chromosomes as they are pulled in opposite directions. Then in telophase, this is where the nuclear envelope reforms. This is also where the division of the cytoplasm happens, but you can kind of just group these together. And this is where the pinching of the cytoplasm happens. So I believe that that's everything for mitosis. Let's just have a quick look. So yeah, chromosomes for prophase. Chromosomes condense. Centrioles move to opposite poles, spindles form, nuclear membranes break down, yeah. In metaphase, chromosomes line up at equator, spindle fibers attached to the centromere, yep. Anaphase, the spindle fibers connect, pull apart the chromosomes to opposite spindle fibers, splitting it into two sister chromatids. And the centromere divides, yes. I think you can also... Yes, you have to say... Sorry I didn't mention this. The spindle contracts. Instead of saying the spindle pull, it's the idea that the spindle will contract in order to enable this pulling apart. Then finally for telophase, the nuclear envelope reforms, division of cytoplasm, pinching of cell membrane. Yep, okay. So that is that. That is mitosis. You could easily get asked an essay question on the importance of cycles, the importance of DNA, anything like that, and you could refer to mitosis, because talking about the importance of mitosis is quite a very broad thing. You could say it's for things like cell growth, cell like repairing damage. A big one could be talking about the idea of, for example, helper T cells undergoing mitosis, allowing for quicker activation of different immune responses. That would be a great example to give in the essay question. This is an interesting question. Apart from... Other students in the class follow the same method but calculate a different mitotic indexes. Explain why. This would be... I always get this question wrong. This is the idea that, like... I mean, what? You're looking at different regions of the tip. So yeah, you're looking at different fields of view. You're looking at different parts of the tip. They're different ages. They're in different conditions. A single field of view does not represent the entire tip. That would be, like, quite an interesting question, I think. Because you can't exactly go wrong with questions like that, where it's asking you suggest why results vary, but you can typically get that question wrong if you overthink. Okay, I'm looking for required practicals, that's all I'm doing. Okay, I've looked and there's nothing here. What about the 2020 paper? I'll take a look at this. Again, we're just looking at required practicals for now. Okay, this is a question that I'll look at anyways, just because it's about immunology. Suggest and explain how viruses became able to infect other species of frog. Okay, so this is talking about viruses have infected a large number of frogs of different species. They are closely related and all belong to the same group. Previously, they only infected one species. Is this the idea? Oh, what could this be about? Is this going to be speciation, or is that too broad? I'm assuming this is the idea of random mutations leading to it being able to bind to... I don't know how you would word that exactly. Literally just random mutations enable the virus to bind to different species. Mutation allows the attachment protein / virus to bind to other receptors. Okay, that makes sense. Name 2 techniques the scientists may have used when analyzing viral DNA to determine that they were closely related. Is this going to be when analyzing DNA to determine relation. Is that going to be like the gene technologies? I'm assuming one would be the gel electrophoresis. I guess just genetic fingerprinting. Yeah, you're allowed to say that. Okay. You could have also just said DNA sequencing. Okay. Determine how the genome of viruses could allow scientists to develop a vaccine. This is the idea that by determining the... wait, it doesn't say proteome. Oh. I was going to say, I guess in terms of genome, you would be able to identify what proteins can be produced. Looking at, like, the antigen structure, possibly? Yeah, okay, identify proteins that derive from the genetic code. Or, okay, identify the proteome. Could then identify potential antigens. Okay. So, determining the genome then allows you to determine the proteome. That's fair enough. Describe how B lymphocytes of a frog would respond to vaccination against the virus. You can assume that they respond the same way as B lymphocytes in humans. Do not include details of cellular response. How B lymphocytes would respond. How are we meant to do this without talking about cell response? I'm assuming you can't mention then that the lymphocytes will divide. So what, you just have to say, what, plasma B cells produce antibodies that are complementary to the viral antigen, and then you talk about, like, agglutination, things like that? Oh no, you are allowed to talk about them dividing. Okay. B-cell antibody binds to viral antigen. They clone. They produce antibodies. And they develop memory cells. Okay. That question kind of threw me off then. Because it said don't include details of cellular response. I wasn't going to talk about mitosis, but it wanted you to. That's interesting as well, that they word the process differently. I just realised, are people able to comment in chat? Because I'm getting like 20 people watching here and no one's saying anything. Am I, did I mess up something? Or are people just not... Okay, maybe I'm going crazy. So that would... Okay, I understand that. It's the idea that the B cells, plasma B cells, will attach to the antigen. Then they will undergo... See, I always assumed that it was the helper T cells that led to them replicating, not the binding to the antigen. So either this mark scheme is different, or... I don't know. That's odd. Anyways, they divide, they produce the antibodies, and then they later develop the memory B cells. Okay. Hello. Okay. So that's it in terms of immunology. That's a bit disappointing for a 2020 paper. Oh, okay. A high concentration of sodium in the blood can affect blood volume and cause hypertension. Use your knowledge of water potential to suggest how high sodium concentration in the medicine could affect blood volume. Oh, this is going to be a really, like, fiddly question. This is, the idea that if there's more glucose in the bloodstream, then... Oh, is it about the kidney nephron, or... Is it literally just the idea that... Hold on, am I overthinking this? It's not about the nephron. No. It's the idea that the bloodstream will contain more sodium, making the bloodstream have a lower water potential. Therefore, body cells, water from the body cells will move down the gradient via osmosis, increasing the volume of water in the blood. I'm assuming that that's what it wants you to say. Yes, it lowers the water potential of the blood. Water would move into the blood by osmosis, increasing blood volume. Okay. I like how my first assumption was it was about the kidney nephron. Because that's all I've been thinking about all day. I'm just having a flick through the past papers. Let's see. Okay, describe the advantage of the Bohr effect during intense exercise. Have we gotten a question on hemoglobin yet? Yes, I'm in year 13. Did we get a question on hemoglobin in paper 1? I don't actually recall. I have no idea whether we did or not. Well, I'll answer this anyways. Because, you know, essay question could get asked about it. It's the idea that... Oh, which one is this again? You obviously have, like, your dissociation curve. Which, that's a very obvious S shape, but it's not going to look like that. It's the idea that it undergoes a complementary... not a complementary, a conformal shift. Oh, okay. The dissociation curve, it undergoes a conformational shift, making binding to oxygen easier. The Bohr effect is where when the... Is it to do with pH? Okay. I'm glad I misremembered. Whoops. Yeah, the Bohr effect is where it's the CO2, because of respiration, it basically makes the hemoglobin have a lower affinity to oxygen, so it can more readily offload oxygen to respiring cells. And my laptop's gonna die. Great. Just a sec. Because I was gonna say, we probably won't get a hemoglobin question. But it could always be worth writing about in the essay. Like, you could get asked, describe the importance of proteins in organisms, the importance of complementary shapes, maybe? Yeah, I guess. Math's more important. Oh, wait, is it the... When is it the next math paper? Okay, again, all I'm doing is literally just flicking through past papers, looking for any, like, obvious questions. Anything on photosynthesis would be fire. Oh, it's Thursday? Yeah, practical... This paper's heavily practical focused. What I was saying earlier, and what I keep kind of thinking, is we only got... We did? I swear we only got respiration, no? Predictions. My prediction for the essay questions, basically what everyone's saying. I hope it's on water. That would be a lovely one to talk about. So much you could talk about for that. Yeah, what I was saying earlier is, what can you talk about for water? Yeah, but that was paper 1, right? So I don't think they would, like, they didn't go into depth about, like, the Calvin cycle or anything. I'm studying for A-level biology. what would you talk about for water? I would go with photosynthesis, translocation, the properties of water in that it acts as like a buffer, and probably, what else would I have done? What's the other obvious one that I've completely forgotten? Yes, osmoregulation, that's it. Wait, did I say it in the Calvin cycle for... I meant, the light-dependent reaction, my bad. But I hope it's not DNA, yeah. Whoops. It is. You know what the thing is with the essay question? My head literally just goes blank. That's why I mean, most of you guys probably already do this, but if you're looking at paper 3, it's probably best to flick to the essay question first, just to see what it's about. So you can kind of be thinking about that. Tissue fluid in... Oh, yes! That's what I thought as well, actually. A couple of days ago. I was thinking about that. Like... I feel like you... Maybe? As long as you heavily emphasized the importance of water in doing that, I think that could work, yeah. I think so. Well, what I was gonna say, sorry, is, in terms of practicals, we only got one required practical out of paper 1 and 2, and that was the choice chambers in paper 2. That heavily suggests we're gonna get 1 or 2 required practicals here. What I think a lot of people, including myself, are thinking is probably a photosynthesis practical. Either something to do with, like, the photometer, which isn't required, but the photometer practical, or a required one on something like the chlorophyll, what the hell? Chromatography for chlorophyll. Or the DCPIP practical. I mean, mate, you basically, yeah. I'd say definitely go for that. If it's on water, you could probably squeeze a lot of importance out for that. Yeah. So yeah, I think in terms of practicals, definitely the nicest one would be the photosynthesis practicals. The photometer one is just investigating rate of water uptake. That's like a lovely one to write about, really. It's basically the same as a respiration practical, which I'm standing by the idea that they're not going to include that practical in paper 3. They included it last year. We already had a question on respiration. I really don't think we're going to get asked anything on the respiration practical. We might get asked stuff on photosynthesis. I really hope we do. Oh, that would be nice. Because you can get up to like 3 or 4 marks for that. Talking about the idea of one being able to... I can never pronounce it right. Metastasize? Yeah, you could easily yap about that. That would be a good one. In terms of other practicals... Hold up. I'm going to pull up... Pulling it up. Investigating simple animal responses. So we've done require practical 10. Possibly a dissection question. They seem to always, like, love the dissections where they give you, like, 1 or 2 marks asking, like, they give you 1 or 2 marks saying, oh, how would you ensure you do the practical safely, and then you can easily get a couple of marks. Yeah, dissection would be nice. The thing is, I noticed they're getting stricter and stricter with those questions. Like, a couple of years back, you would get, like, 4 or 6 marks for a dissection practical. Now it's, like, 2 marks. They seem to be like toughening up on those like loose questions. Things like the chromatography practical, you can always get marks because they always kind of ask you the same thing. It's like, why would you draw the baseline with pencil? Why would this solvent be more like why would there be more pigments with this solvent than the other one? And it's kind of the same stuff every time with the chromatography practical. Um, the DCPIP practical, that's the one that my teacher thinks um, would kind of pop up on. I think, have you, the unofficial ones? I have not seen those. But, honestly, looking back, like, I definitely made some obvious mistakes with Paper 2, which is gonna, like, haunt me for the next couple of months. Dehydrogenase activity in chloroplast, that is the, DCPIP one, right? Yes, okay. The DCPIP practical, I do hope it comes up. Because they can ask it in a variety of contexts. They can ask it based on, like, light intensity, temperature, anything like that. And they usually ask kind of the same things. Like, you'll notice with a lot of practicals, they'll always ask you, like, one or two questions. Oh, I hope so. I really, really hope so. Yeah, I'm in the same year, dude. I'm not a teacher at all. I would dread of being one. Yeah, my honest hope is that photosynthesis comes up, like, one or two questions. Better than that than right, buddy. Stop saying that. That's lies. Okay, one that I've just thought of is aseptic techniques because we have not gotten a single immunology question. Yeah, we got asked stuff on, like, cancer. We got asked something on bacteria in relation to plasmids. We haven't gotten anything on the different types of immune response, vaccines. I would definitely hope that immunology comes up. Because being able to talk about aseptic techniques, that's usually like 4 or 5 marks. And it's literally just saying, sterilize the equipment, work with a convection current, sterilize your equipment, disinfect surfaces, flame the neck of a bottle. Like, it's the same stuff, but you always get marks for it. Wait, really? Last year there was none? That's kind of interesting then. That is interesting that they haven't included it last year. We did, but I think that linked more to like natural selection and because antibiotic resistance, yeah, that's more to do with like the like populations and stuff. I hope a question on speciation comes up. Speciation is such a nice one to talk about because whether it's geographic or reproductive isolation, it's the same thing. It's the idea that there's no gene flow between the species, leads to variation due to mutations. You can easily get a lot of marks out of that just by saying the same stuff, which I hope. I would love that. Um, I'm just kind of flicking through my notes, seeing if there's anything. Obviously, what a lot of people I know are dreading is the gene technologies topic. Yeah, you sometimes get a mark literally just for saying geographic isolation. Like it's a lovely one, and I've seen in the past papers where you can get so many marks just for talking about that process and linking it to the question. I hope gene technologies doesn't come up, honestly. Like, whoa, the sky is blue, but yeah, I hate that topic so much. The only thing I feel fairly confident with is the polymerase chain reaction. But everything else is like a big no. I wonder whether we would get tumor suppression, like a question on tumor suppression, given that we got a similar question in paper 2. I wonder. Probably not, because we did get a big section. Thing is, it kind of did, yeah. Like, the idea of the gel electrophoresis identifying cystic fibrosis, I would pray that means it doesn't come up again. Because, yeah, I might be fucked if that comes up. I just have to start making some stuff up, honestly. What else? Obviously, respiration came up, so it's highly unlikely that's coming up again. Photosynthesis didn't. Nitrogen cycle did, which I'm glad. Honestly, I'm a bit disappointed as to the lack of, like, action potential questions we got in paper 2. Things like, photoreceptors, pressure receptors, that didn't come up. Like, you could get a massive section on, photoreceptors. What an immunity. Honestly, like, they could ask a lot. Like, the most obvious thing to me would be aseptic technique, because then they can link it to a practical. It's probably going to be something about, like, a specific response, like, investigating the response of B cells. Yeah, this is live. Because then, the thing as well is, paper 3 is heavily focused on the evaluation questions, where they want you to talk about, like, the issues with certain, like, conclusions, things like that. I love those questions. The evaluation questions are lovely. You can basically milk marks out of it, like, really nice. Like, obviously it's the idea of, you get given, like, a bar chart, and if the standard deviations overlap, then that tells you that, the data is not statistically significant. The idea of p-values as well, that's a big one. Talking about the idea, although I realised we did get asked a question about p-values in terms of, like, the chi-square test. I don't know whether they'll ask us a chi-square question again. I feel like probably not. siRNA and oncogenes. Honestly, that's the one thing that I hate. I could try. I could try in a little. I know siRNA is just, that's the one where it, like, limits translation? Honestly, I don't think that process is too bad, actually. Hold up. From what I have written down in my notes, it's the idea that it associates, you have your double-stranded, which, again, crude drawings, please bear with. It's the double-stranded siRNA. It associates with a splicer protein. Then it becomes single-stranded. It binds to a target mRNA because it's complementary. So they're complementary. And then it literally just cuts the siRNA into smaller fragments. Because siRNA has proteins on it that will prevent it. What does my name stand for? Oh no, I'm not a teacher, by the way. I'm aware I'm probably, like, the only person in the universe sad enough to revise biology by streaming it, but I am not a teacher. I don't know what I'm talking about. Okay, what do people think about enzymes? Because according to my teacher, she thinks it's very unlikely we're going to get asked more enzyme stuff, given how much came up in paper one. So that leads me to believe we're not going to get a question on the first required practical. Feel like that's unlikely. Mitotic index could come up. Yeah, but honestly, that's what I found. Like, it definitely is. Like, I've been doing these since GCSEs. Like when I started GCSEs, like in year 11. I think it's been really helpful. Like, I find the best way I revise is just by like being able to explain it. Oh, random sampling practical would be lovely. Quadrats or the capture recapture. Either of those would be lovely because you always get marks for saying the same like two things. Like for quadrats, it's literally always just a method of randomly obtaining coordinates. Literally just saying using quadrats. And then usually a mark for talking about the large sample size. The photosynthesis prac. Yes, I love that one. I love that one so much. That's a lovely practical. Yeah, the essay is probably the worst bit. Like, thing is though, realistically, like, I don't know how you guys revise for essays or anything. The way I do it usually is I have a handful of processes that I always can just remember right off the top of my head. Things like co-transport, for example. You can link that to so much, right? Like you could link co-transport to the role of proteins, because talking about the channel and carrier proteins, the role of diffusion. You could talk about active transport or ATP. Like, co-transport is one that I use all the time. The role of ions, like, that would be... Co-transport's are one I always write about, because you can apply it to so much. Another one I always talk about is translocation. You can apply that one for water, active transport, ATP, not really osmosis. Possibly you could apply it... Don't know what other processes actually. But that's still like a big enough list. The April Fool's video. Thank you. I'll type out that guy. Right, yeah, like pressure gradients as well. And if you really wanted, you could even maybe... I think also you could talk about, like, the interaction between cells. Because you have the companion cells. So again, that would be a great one. The other process I love to talk about is, like, synaptic transmission. That's a lovely one, because again, you can talk about diffusion, the role of ions, the role of channels, the role of complementary shapes. So many. I hope the kidneys come up. I only do 2 A-levels, actually, which is kind of mad, but it's because last year I did core maths, this year I've done extended projects, so I don't even have 3 A-level subjects. That's, which is kind of sad, but I do history and then biology. Yeah, I hope kidneys comes up, I really hope so, because kidneys, like, the whole topic of that is lovely. Osmoregulation, lovely. Influx of calcium. I think the way that I have it, at least, is it's when the action potential arrives, that causes the sodium channels to open. Obviously, sodium will diffuse through. That causes the vesicles to fuse with the membrane, releasing the neurotransmitter. So I think it's just the arrival of the action potential. Oh, dude, you have my you have my condolences. That's awful. You definitely can't get into uni, man. Like, you gotta think about it this way. Like, there's some people I know that like could not care less about A levels. There was some guy that showed up half an hour late to my history exam. Like, you gotta think at the end of the day, you know, you guys are putting in more effort than most. Let's see. Sorry, I missed a comment earlier. If a question asks importance of ATP, yeah, usually what I would do is if a question was like, oh, discuss the importance of ATP, I would talk about something like co-transport, and then you obviously explain the role of ATP in that process. The role of ATP being that it enables the sodium potassium pump. Kind of talk about that. You need 70. Oh, dude. That's unfortunate. Like, that is unfortunate, dude. Yeah, exactly. Like, at the end of the day, like, it's the effort that counts. And I genuinely wish all of you guys the best of luck. Like, who is this guy, bro? Sorry, the reason I literally can only see like 2 messages in chat at a time, so that's why I'm being a bit of a prick. Okay, the other practical I was thinking as well, the diffusion and osmosis practicals. I don't know whether they would come up. Was last paper's question about increased respiration? Why does the pH decrease? Isn't that the paper 1? Where they asked about it in relation to, like, pairs. I thought that was because of the hydrogen ions. And, like, the increase of them led to the decrease in pH. What am I doing? I'm losing it. Yeah, that's what I thought. Man, I'm actually losing it. Like, just closed all of my tabs. God. This is what biology does, man. My head is killing. Can't wait to be done. Cannot wait to be done. Increase respiration. Wait, for paper 2. I'll be honest, I cannot remember. Negative. Oh, that question. Yeah, I think it was. Yeah. That's what I put. I remember now. I remember. I think that, yeah, because obviously respiration, that would decrease the pH. Yeah. Yeah, I think that's right. Honestly, I probably fumbled that question on negative feedback. I probably did, I'll be honest. I think I fumbled quite a few questions, but I don't know how many of you guys do it, where you count how many marks you think you've got. Like, at the end of a paper once you've gone through it all. Obviously, it's a bit hard to do with paper 3 because it's the essay question, but that's like my go-to thing with this paper, biology. Okay, what I'll do is I'll quickly have a look through 1 or 2 more past papers. I kind of run out of papers, honestly, I've run out. I'll look at the specimen paper, which is like, this is the unholy grail of biology. This is horrible stuff. The 2014 specimen? Oh, hell no. Honestly, I've probably got like, I think for both, I got around like 60 marks. But again, just complete like rough guess. Like, the way I usually do it is I'll count up how many marks I think I've got, like, worst case scenario, so I end up, like, knocking off a load of marks for things that I probably got right, but, you know. And then I kind of do one where I think I've gotten more right. So it's about a 50-50 assumption. Okay, well, I've looked at... There's a question here. Talking about the mark release recapture method. They used a fluorescent powder. They captured insects from a number of sites on the island, suggest how they decided where to take their samples. That's a lovely question. Just talking about random sampling, like generating coordinates. Give 2 assumptions that must be made when using mark release recapture. Again, that's a lovely question. Talking about the idea that whatever you're like tagging them with can't affect their chances of survival. And also that you have to assume that like the birth and death rate remains constant. That there's no immigration, nothing like that. Because obviously, if the population changes, your estimate's going to be wrong. Oh, but did I say immigration, bro? Actual brain rot. Suggests the advantage of using fluorescent powder in this experiment. That's the idea that the fluorescent powder is not going to affect survival, because it only reacts to UV light. Okay. Use the information provided. Suggest one way in which the method used by the ecologist might have caused an increase in their estimates. Is that by using a small sample size? Okay, yeah, this mark scheme is like horrible. I don't understand a thing that says. They removed large numbers of insects which were not returned to the same area. So it affects... Oh, okay, I get that. I hope... I mean, nitrogen cycle already kind of came up in that final 15 marker for paper 2. Okay, what an odd question. Describe how you could make a temporary mound of a piece of plant tissue to observe the position of starch grains in a cell. That is an odd question. I'm assuming that's literally just basic microscopy though. Like the idea of adding a stain, using a thin layer so that light can pass through, adding water to the slide. That should be fine. Okay, they won't ask this on paper 3, surely. Biological drawings. Because they seem to be very infrequently asked. Questions like where you have to draw graphs or draw tables as well, like, it seems like they ask those very infrequently, but I hope those don't come up. Ow. Just cut my nose. Let's see. Okay, I notice as well, there seems to be a handful of questions where they ask you really basic math stuff. Like, a 2 marker here, how would you find the median survival time? And it's literally just saying a range from smallest to largest, and it's the middlemost value. Obviously, the whole gimmick with paper 3 is there's a lot of evaluation questions. Questions where, you need to talk about the limitations to a conclusion. I love those questions. You can normally always get a lot of, like, easy marks off them. They're usually really nice. So again, in terms of what I honestly think will come up, and again, I'm not like a prophet, no idea. My honest predictions is probably an immunology section, possibly linking it to a practical. I think definitely photosynthesis is going to come up in some capacity, possibly a practical. I feel like we'll either get a section on osmoregulation or muscle contraction, because it seems like such a broad topic to not include. Like, muscle contraction is a massive, like, talking about the sarcomere and, like, the different bands. Maybe receptors comes up again, like photoreceptors, mechanoreceptors, like, I mean, homeostasis did come up, but not to much extent. Like, even thermoregulation didn't really come up much. Regulation of the cardiac cycle did, but that was about it. Again, if you got asked an essay question on the role of receptors, the role of receptors would be a really nice one to talk about. Because again, role of receptors, you can link back to a lot of things. Okay, again, just kind of flicking through this paper. A lot of questions have been stuff that's already come up. Oh, hello. Welcome. Man, honestly, these streams are, like, mad in terms of the people that we draw in. Like, for context, again, I've been doing these since GCSEs. So I've been doing these streams for about 3 years now. And yeah, like, it's mad. Because most of the time these get, like, 0 views. I'm assuming because it's exam season, everyone's panicking, and now we're getting a lot of people in. But honestly, like, it's chill as hell. You guys are all cool as hell for watching. I appreciate it. Man, I can't really think of anything else now. My head's kind of gone blank. Probably a sign that I need to stop, but we'll go through 1 or 2 more things. Methylation. This is one that I thought of. I don't think methylation's a particularly bad one. It's literally just the idea that when you... In fact, no, this won't come up. We've already done it. Ignore me. Transcription control could come up. That could be possible. Let's have a look. Obviously, the DNA technology is, like... I'm not revising these. I'll just, It's like the role of, restriction endonuclease. I don't know how much detail you have to go in to get marks for this. I don't know. The only thing that I have is that they cut hydrogen bonds in the DNA. They form the sticky ends, which are pieces of DNA that just have exposed. Okay. I'm saying immunology, synaptic transmission. Yes, I hope. Photosynthesis would be lovely, man. Like, yeah. Kind of, we're basically on the same wavelength. Like, immunology is definitely has to come up, surely. I'm predicting water. But honestly, talking about it now, like, we might be safe with a lot of things. Like photoreceptors. I want to quickly go over that. Photoreceptors is literally just the differences between the rods and cones, because the process itself is relatively easy. It's just the idea that the pigment gets bleached. Obviously talking about the bipolar neurons, the idea of that. It's the rods have a high visual sensitivity. And that's because obviously the rods, multiple rods are connected to the same bipolar neuron. Therefore, there is enough neurotransmitter to always reach the threshold. You can refer to this as a lot of things, like, obviously it's retinal convergence. Or you could just say spatial summation. Remember, there's spatial summation and temporal summation. Bro, what are you on about? Photosynthesis was not in paper 2. That's mad. Cones are the opposite. They have a high visual acuity. And that's because each cone cell, again, ignore the crude drawings, each cone cell is connected to its own bipolar neuron. Therefore, the cone cells send separate impulses to the brain. So again, it's this idea that in rods, it's enough neurotransmitter to reach the threshold. With cones, it's separate. Myelinated neurons came up, so I don't need to go over that. What else? That was respiration. Because it was... Wait. No, that was the first question, which was respiration. I swear the second question wasn't on photosynthesis. Yeah, the special is, hold up. Special is where there's multiple, synapses, so there's more neurotransmitter. Temporal is, I think, just an increase in frequencies over time. Like, in a shorter space of time. Could potentially get a question on that, to be fair. Again, like, there's a lot of comparisons, but again, with paper 3, it's more stuff like evaluation questions, practical questions, things like that. Again, speciation, I would love to come up. Intraspecific and interspecific competition, that would be nice. Intra being between the same species, inter being between different species. Again, action potential, that would be a lovely one, because you could, like, there's a lot to talk about there, which is why you could easily get asked like 5 or 6 marks for that, potentially. Again, response to stimuli with the Pacinian corpuscle, response to stimuli with photoreceptors, synapses, I hope comes up. Sliding filament hypothesis, that I would love that to come up. What are your essay? The ones that I hope what I hope comes up, water would be a lovely one. Again, I've had this planned in my head for a good while now. You could talk about the properties of water, the idea that water acts as a buffer, requiring a large amount of energy to raise the temperature, you could easily talk about the importance of that property in relation to things like enzymes, preventing them from denaturing. That would be a lovely way to get like the AO2 section done. In terms of other things, I would go with translocation. Obviously, the role of water in that process, establishing the hydrostatic pressure, establishing the gradients. I would also go with the light-dependent reaction, talking about photolysis, the role of water being split, providing the electron to the chlorophyll, while the hydrogen ion establishes the gradient. And I would also go with osmoregulation. Because obviously then you could link osmoregulation to the idea of water, the idea of the water level being maintained so that cells don't crenate, things like that. That would be what I would go with, anyways, if the question was on water. I don't really know what else could come up essay-wise. I mean, things like the importance of DNA, like, you know, you could stretch out a couple marks for that. The importance of cycles would be a lovely one, I feel. Talking about things like the nitrogen cycle, meiosis, increasing genetic diversity, things like that. Obviously, the Calvin cycle and the Krebs cycle. That would be a lovely one. Energy transfers in and between organisms. I don't remember what I did for this one. I think I talked about the... Oh my god, I don't even remember what you call it now. The trophic levels. I mentioned that as one point. Don't know what else I talked about, to be honest. Because we've done a lot of these essays in class, that's what I'm remembering. I think I mentioned the trophic levels. The idea of like conserving energy between levels, things like that. One of them I talked about was, it might have been synapses, possibly. Because you can talk about like photoreceptors, mechanoreceptors, because that's energy transfer. Same with, I spoke about the light-dependent reaction. I know that's what I did. Because obviously that's like, you know, photons being converted into ATP, things like that. I don't know what else to talk about for energy transfers. I guess if you wanted to be really generic, you could talk about ATP acting as energy transfer in the organism. The role of ATP in that context. And then the role of energy transfers between organisms, I would not know what else to talk about there, to be completely honest. Maybe like digestion? I don't know. Could I would be stumped with that question. Energy transfers in and between organisms. Obviously that's the in, that's the in, that's the between. I don't know what else you could talk about. I guess maybe the nitrogen cycle. I wonder, I'm probably gonna wrap up. That's kind of why I'm being a bit lazy now. Probably gonna wrap up the stream in a sec. So, overall, what I will leave things with for now is I definitely hope there's something on immunology. I hope something on photosynthesis. Possibly for both of these practicals would be lovely. Things that may come up but may not. Feel like osmoregulation might come up. Muscle contraction might come up. And speciation, I hope that comes up. In terms of essay titles, my personal one is I hope water comes up. It's probably going to be something really shit that I'm going to run out of ideas for halfway through. In terms of other things, in terms of practicals, I hope the chromatography practical comes up. Or the DC PIP practical. Those would be lovely. Maybe a random sampling practical. And as well, maybe a biochemical test. Like the test for like proteins, the test for reducing sugars, non-reducing. That would be lovely. That would be a lovely one. But that's basically it, honestly. Those are everything for my predictions. That's what I'm going to spend the rest of the day revising. And yeah, thank you guys for watching. This is now literally, this is it now. After this, I will not be, active on this channel anymore. Thank God. But yeah. That's basically it from me. Good luck, everyone, obviously, with the paper tomorrow. Feel free to put in the comments what you thought of the paper, stuff like that. I'm doing biomed in uni, so I'm not escaping biology, I'm just escaping the A-level revision. Which probably is going to get a lot worse, to be fair, but yeah. But yeah, good luck, everyone. Man, we've got 300 people who have watched this stream. Thank you guys. I hope all of you watching do well. And yeah, literally, I've been doing these revision streams since GCSEs. And that's basically it now. That's me done with these streams. Good luck, everyone. And yeah, goodbye.